Good morning. So, today I have a very interesting topic for you, that is personalized medicine and holistic healthcare. So, when I talk about the term personalized medicine, it itself defines that medicine which differs from person to person. This medicine can be food or drug. We all are very different in the genetic makeup that we all know. But do we also know that we have a very distinct pattern of microorganisms in our body and which is very unique to every individual, and this is like a fingerprint to that individual. So we cannot focus on all the microorganisms in this talk. So today I will be focusing on the microorganisms that are present in the gut of an individual and how they have the correlation with our health and disease. So if I talk about the holistic healthcare system, then I have to take an account of some important terms like how I can get a holistic means a whole healthcare that involves all the systems of the body, that is the gut, the mind, and other parts of the body. So that is called as the holistic healthcare. So this will involve so many molecules, like suppose if I take about the microbiome, then I have to monitor the biomolecules that are synthesized by these microbiomes and the role of these biomolecules in our metabolism. The second is the functional nutrition. These days there are so many deficiencies that are occurring in an individual, so we need to take an account of all those deficiencies and replenish them by the help of food. And the functional nutrition is dependent on the type of body microbes that a person has. The gut microbiota that he possesses defines what type of food he should be advised in this functional nutrition. Then here we take an account of another condition, that is the detection of a leaky gut. Leaky gut, I will discuss in the subsequent slides what is a leaky gut, but just briefly stating that in this condition there is the permeability of the gastrointestinal tract, it increases and it absorbs many of those molecules which are unwanted and which may act as toxins to the body. Then the other is, if we take the mind-body correlation, then there are other therapies which can be helpful for the mind, like meditation, sound therapy, music. The next part is how we can balance the gut microbiota and also heal the leaky gut condition to get a better digestion and absorption of the nutrients. So these next part is detection of these microbes with advanced tools. So nowadays there are many tools that are available for the detection of these gut microbes. There can be others like Raman, which is very specific diagnostic tool and which no one is working on for the diagnosis purpose, but this can be developed and it can be a very precise way of diagnostic of even a single cell. So taking into account all these, we can actually detect and also suggest that what diet one should be following and what diet one should be avoiding, so that the symptoms of the health conditions, they disappear slowly. Next, talking about the gut, Hippocrates said that all diseases begin in the gut and health is determined by the microbiota in our gut. So this seems to be a very important comment, that all the diseases and Also, the health is determined by the gut. So, let's see what is a leaky gut. So, these are the intestinal cells, and here these are structures are called as the tight junctions. So, in a condition called leaky gut, these junctions become more porous, and the absorption of the toxic molecules into the bloodstream take place, and these in return can cause many of the consequences and also may cause autoimmunity to an individual. So, what are what there are some agents like gluten, casein. So the proteolytic effect of these molecules may cause a condition called as the leaky gut. So these like gluten is found in wheat, and casein is a molecule that is found in milk. Another is if there is a dysbiosis in the gut flora. As I talked about, that gut microbes are very important, and they provide a very ambient atmosphere for the assimilation and digestion of the food. So if there is an imbalance in this gut flora, then also this leaky gut condition can take place. Stress is a very important factor which in today's life almost all of us are undergoing, and low fibers in the food. So all these are some of the reasons of a leaky gut situation. Then they can be aggravated by medications, long usage of medicines like aspirin, steroids, antacids, antibiotics, etc. So because of these reasons, there can be a low grade inflammation in the gut. So one of the common problem is IBS, that is called as the irritable bowel syndrome, and this can is related to leaky gut. So this can happen if there is a change in gut microbiota. This leads to low grade inflammation, and in this condition we can see that the bifidobacterium and lactobacillus are reduced in the irritable bowel syndrome. So causes of leaky gut, as I already discussed: stress, imbalanced microbiota, medications, toxins that we take with food, and other pesticides and all that we take, then poor diet. So when should we get a leaky gut test done? That is, these are the conditions enlisted that a person can associate with leaky gut condition, that is the food allergies and intolerances, the autoimmune conditions like diabetes, irritable bowel syndrome, digestive problems, skin diseases, like if there is a brain fog, confusion, headaches, mood swings, depression, excessive fatigue, and if there are no nutrient deficiencies, then we can first think that there can be a situation of leaky gut. That is why the absorption of the nutrients from the intestine is not taking place properly, and so the detection of this leaky gut condition is very important in all these symptoms and diseases. So one of the tests that is used for the detection of the leaky gut is called as the zonulin test. So this zonulin is a protein, and this protein is increased in the condition of leaky gut, and this zonulin level increase when the gliadin level increases, and gliadin is a protein that is found in wheat. So there can be a direct correlation between the consumption of wheat and the levels of zonulin in the blood, and if this does not suit an individual, then this leaky gut condition can take place. So as I already said, that how the leaky gut condition can be diagnosed by the blood test called as zonulin. There is another test called as the mannitol lactulose test, in which the urine is collected after 6 hours of ingestion of these two sugar molecules. So high levels of mannitol and lactulose in the urine is indicative of a leaky gut syndrome. So after 6 hours, if we see high levels of lactulose and mannulose in the urine, then we can think that okay, this person may have the condition called as the leaky gut, and other confirmatory tests are again done to confirm the symptom. So if we have to see the health of the intestine, then these are some of the tests as I already mentioned. So if we see, this is the structure of the gut. The gut, this is the gastrointestinal tract, and here you see there is abundance of microbes in different regions. You can see different types of microorganisms are present. So some may be harmful and some may be useful to the host. So an estimation of all these in the gastrointestinal tract is very, very important to estimate the health of an individual. So just to define the terms microbiome and microbiota, some people may get confused what is a microbiome and what is a microbiota. So microbiota means it's the sum total of all the organisms that are present in a specific environment, and microbiome is taking into account not only the microbes but the genes and also the genomes of these in a particular environment. So since the birth, the microbes start colonizing in our intestine, and not only since birth, but since a child is in the uterus, and then after birth this process again starts, and you can see there are reports where they have shown that a child who is born with a cesarean delivery and a child who is born with a normal delivery have different composition of the gut microbiota, and this is a very important finding, which also suggests that of course the conventional route of delivery is the best route, that because that takes into account the microbes that are present in the female genital tract and is very good for the colonization of the gut microbiota in the child. So if we talk about the gut microbiota, we have bacteroidetes and firmicutes. They make 90% of the gut microbiota, and each individual harbors his or her own distinct pattern of gut microbial communities, which I already discussed earlier. Okay, and for a given individual, the fecal microbiota remains almost stable over a person's lifetime. So the colonization starts, and the fecal microbiota after a certain period of time remains remarkably stable and becomes the fingerprint of that particular person. So there are some factors that influence the microbiota, like it changes with age, geography, diet, stress, and medication use. So what are the functions of these gut microorganisms? That is, they help in developing the immune system, then in developing many molecules that may be useful for the metabolism of the food or for the nutrition of the body. Okay, so very vital molecules like vitamin K and folate are also synthesized, and the gut microbes have a very important role to play in the synthesis of these vital metabolites and vitamins. Then in the drug metabolism, they play a very important role. They deconjugate the bile acids. So these are only some of the functions enlisted for the gut microbiota. So what is dysbiosis? If there is an imbalance in the composition of the gut microbiota, then we call this condition as a dysbiosis, and this condition may lead to several imbalances in the metabolism, and a person might develop many metabolic disorders over a period of time. So, to estimate these microbiota or the microbiome of the human microbiome project was done, and this is a very big chapter, a very complex thing, so this cannot be completed in this particular lecture. I may have to take another lecture to just explain how and what the microbiome project was about, what were the objectives, what were the observations, and what were the experiments that were done. Then how we can study these microorganisms? So there are three ways to study. One is, you can see this microbial community can be studied by three major ways, that is, what microbes are present, on the basis of what they are doing, and what is the genetic potential. So which microbes are present we can know if we do the nucleic acid analysis. Then what they are doing is, if we see that the expression of the RNA, the proteins, and the metabolites. So RNA expression is metatranscriptomics, the proteins is the metaproteomics, and the metabolites is called as metabonomics. And if we see the genetic potential, that is the DNA of these gut microbes, then it's called as the metagenomics. So another important element of the gut microbiota is the virome. Virome means the viruses that are present in the gut microbiota and their role in pathogenesis and diseases. So this I will discuss in another lecture in a very detailed way. So another aspect is the human mycobiome. Myco means the fungi. So here you can see that right from the mouth to the colon there is a distribution of these microbiota, and also you can see the variations of these microbiome with the various parts of the body, like the oral cavity, the lungs, the gastrointestinal tract, the skin, and the ones that are enlisted in red show the pathogenic species that are inhabited in those areas. So there can be some bioinformatics approaches for understanding these microbes and what microbes are present. Okay. So the most important, the most conventional, I should say, way of detection of the bacterial species is done by the 16S rRNA sequencing. So 16S rRNA is a component of the ribosome of the bacteria, and this sequencing is done for the identification of the bacterial samples. So this will take another lecture to explain how this 16S rRNA sequencing is done for the identification. So these are the steps involved in the 16S rRNA sequencing studies. So there is a company called as Viome. So Viome is doing similar work in detecting the presence of the microbiota in the gut of an individual, and also suggesting them what food they should be eating and what food they should be avoiding on the basis of the microorganisms that are present in the body. So this company is set up by Naveen Jain, and he claims that he can actually give suggestion on how to improve the beneficial microbial species. Then if something is missing, if some class of microorganisms is missing, then he can actually replenish with the help of the probiotics. Then whether a person is lacking any good metabolite, and which prebiotics are good for an individual, and what should be the ideal ratio of the proteins, carbohydrates, fats that a person should be taking in the diet, and other dietary recommendations are given by Viome. So, this Viome uses a technique called as metatranscriptome sequencing. So it looks into not only the RNA but the expression of the RNA and metabolites in the gut, and it compares his method with the 16S rRNA sequencing method that is used conventionally for detection of the microorganisms. So Viome claims that they can identify all the bacteria and all other living organisms in the gut, whereas the 16S can only identify a fraction of these microorganisms in the gut. And Viome can detect species and strain of the microorganism. The other is that it measures the biochemical and the activities of the microbes, which the 16S cannot identify. Then the results that he obtains by this method has less variations. He is able to identify the missing metabolites in the gut of an individual. And so basically Viome gives a picture of the complete picture of the gut health and the dietary and lifestyle changes that a person should be doing for the improvement of the gut health. So if we talk about in India, national level, then a company called as Leucine Rich Bio is doing similar work. So they have created a diagnostic tool called as Bug Speaks, and by the help of this diagnostic tool they can estimate which microorganisms are present, and also they are able to predict the susceptibility of around 20 diseases, and then basing upon that they can provide the nutritional and probiotic recommendations for an individual. So we are collaborating with this company for the study of the gut microbiome of the pediatric patients, and we are also collaborating with several hospitals to identify the health of the gut in the pediatric patients. And this study seems to be very interesting because on the basis of that, many of the issues of the pediatric patients can actually be resolved just by the help of the right food. So just to summarize how we can have a healthy gut, because as we have already seen, the gut is the center, the epicenter of whatever is happening in our body. So it's very important that the condition of the gut be first estimated, and then we can find the ways to improve the condition of the gut by the help of the diet, the antibiotics, if there is a certain pathogenic organism which should not be there for the good health of a person. Then prebiotics are the non-digestible substances, that are present in the gut, and probiotics are the living microorganisms that are present, and symbiotics are a combination of the pro and the prebiotics that are given that can be beneficial for the patient. So these are actually some of the recommendations to have. Another is called as the fecal matter transplant. If a person is missing any vital microbiota, then the fecal transplant can be done to replenish the same. So these are some of the ways by which the gut microbiota can be replenished. So therefore, we must always not think that the gut microbiota is causing diseases to an individual, but these gut microorganisms are very important for our overall health. It not only has correlation with the digestion, but also has direct correlation with the state of the mental health. So a person who is undergoing depression, anxiety, mood swings, fatigue, stress, can have a direct correlation with these gut microorganisms. So there are certain lifestyle disorders prevailing these days that do not take into account the health of these microorganisms, but after the study we can see that there is a direct correlation between these lifestyle metabolic disorders and the health of the gut. Gut being the center of everything is very important for the digestion and assimilation of the food that we eat. So these microbes that are present need to be estimated to see if any vital microorganism is missing from our gut, and there can be a correlation between these microorganisms and our present health conditions. So I suggest that all should take care of their gut, if whether it is leaky gut or if there is an imbalance of the microbiota that is present in the gut, to have an idea of the health and the diseases and the consequences of these diseases. So take care of your health and the microbes that are present in your body. Thank you.